All right, let's move on to paracrine factors here. So paracrine factors are um, the longer range. I say longer because endocrine factors or hormones, for that matter, are long range. These are local but can span multiple cells, I guess is the best way to put it. So we're going to talk a little bit more about them because we talked about the juxtacrine factors in the adhesion. So this kind of depicts like okay, except the difference here. So these would be like paracrine factors or the ligands that are diffusing across. Generally, though, what would happen is like if this is the cell, they would bind to membrane-bound proteins. They don't actually diffuse across. This looks like either they're coming out of the nucleus or they're going into. I guess this could be the cell that's excreting them. But it would, if this was a cell receiving them, they would bind to membrane-bound proteins and cause a chemical cascade. If they were excreting them, this would be a little better. They wouldn't diffuse. They'd exocytose. But anyways, we're not going to delve too much into these inaccurate pictures, but they're fun to look at. So remember, we have juxtacrine interaction, and those are membrane proteins on the cell surface that interact with the receptor proteins, so they have to be touching. Paracrine factors, so proteins are synthesized by one cell, and then they are excreted and diffuse over a small distance to induce change in neighboring cells. So this is about 15 cell diameters, right? So like I said, local, not like across to your body, like something that would be like a hormone. And then there's autocrine interaction, is a type of paracrine interaction where the inducer cells create the paracrine factors, but also respond to those same factors. So they they create these factors and then respond to them within itself. So you're not always signaling other cells. Sometimes you create it for yourself. So morphogens can be broken down into 4 families based on their structure. So we have FGF family, which is the fibroblast growth factor. The hedgehog family, funny story. Hedgehog family, originally there was a gene back in the 90s that was discovered and characterized, and it was named Sonic Hedgehog because the developers or the discoverers really liked the game Sonic the Hedgehog. So there are some funny gene names back in the day. They've kind of gotten away. There's been conventions put in place about naming genes, but at the time there wasn't, so that's where hedgehog comes from. There actually is also, they could justify it because the phenotype of the hedgehog was also pointy, so they found a way to justify it, but they've later come out to say that Sonic the Hedgehog was a big influence on naming. There's the WNT family and then the TGF beta superfamily. And this superfamily consists of TGF beta, the activin family, the bone morphogenic proteins, nodal, and some other ones. So it's a big family that kind of gets grouped together. So these morphogen paracrine factors regulate gene expression, and the concentration of them often determine cell fate. So if you're in an area that has high, and we talked a little bit about this earlier, I believe it was in the syncytial differentiation, like these gradients. So if you're in an area that has high concentration of some morphogen or protein, your information is different than a place that has low. And so this information of how much of some protein or ligand or whatever is near, that gives you positional information on what you should develop to. So it changes the number of receptors that are bound on your membrane changes, and therefore your gene expression changes, etc. So cells that experience high levels of the morphogen transcribe genes differently than the ones that have low concentrations. So these paracrine factors function by binding a receptor, and it can cause 2 different things. So it can change transcription of a gene in the nucleus, or it can regulate or change or remodel the cytoskeleton. And this would allow cells to migrate, right? So you can change your kind of binding and things of that nature. And what, remember, those cadherin molecules are attached to catenin, which is attached to the actin cytoskeleton. So you can remodel that and allow cells to migrate, which we'll get into cell migration during the gastrulation stuff. So in this experiment here, this kind of shows like the position or the distance of paracrine factors and how they can travel, right? So they had a 3 different conditions here. So they had beads that have no activin, which is a paracrine factor, and so they put them in a bunch of unspecified cells. So these haven't gone through specification or differentiation or determination or anything like that. They're just kind of stem cells right now. So they put nothing in there and they get no activation or no change in cell type. Then they put beads with 1 nanometer or nanomole of activin, and this caused those nearby cells to start expressing XBRA. Then they put beads containing 4 nanomoles of activin, so a high concentration, and that caused some cells, the outermost cells, to express this XBRA. And then the inner cells started to express goosecoid. So this appears then, we can map this. So if we turn this on its side, right? So we're looking at this kind of from the side point of view and mapping. We see that here's our activin bead, and this is for this last one right here, right? So the high concentration here, if we mapped it back to our previous slide, we see that if you're close to it and you have a higher concentration, then you're going to express that goosecoid. If you are a little bit further out, like towards the ends of how far it'll diffuse, then we'll express this XBRA. And if you're too far away, or where you're getting a none or just a very low concentration, like below the threshold needed for XBRA to express, then you express nothing, right? So this concentration, so paracrine factors are very good at building this concentration distance and helping measure distance, so to speak, or developing distance. So we're going to get a little bit more into each pathway coming up, but we'll quick do an example of a WNT pathway. So in this pathway, packaging the beta-catenin destruction apparatus into endosomes is what we're doing. So exocytosis is one mechanism used to eliminate a receptor at the membrane. So if you were, remember, if we want to leave the cell, or the cell wants to move, and you have these cadherin things, one thing you can do is you can use exocytosis to get rid of those membranes, those membrane brown proteins, so like your cadherins and things like that. And so when WNT binds to its receptors, the beta-catenin destruction complex, which is this DC, that binds the receptor, and the entire complex is internalized into this endosome. So what happens is WNT will bind to frizzled and disheveled, which are wonderful names for proteins. It'll activate this destruction complex, but then internalizes these external, or these transmembrane proteins. So it used to be that these were across this frizzled, disheveled transmembrane protein was across and exposed to the outside, right? But now we've endocytosed and put them inside the cell, and then we get an endosome that comes around, this multivescular endosome, and swallowed up even further into an endosome here. And so this then allows for that cell to not have those transmembrane proteins. So if you were to migrate, or if you needed to dissociate, like I said, we'll get into the migration in a little bit. That is what you can do, and it can actually target for degradation, and they will excise the beta catenins, and then get rid of the rest of the proteins through protein lysis. But don't worry so much about that. But this is way talking about. We've talked a little bit about removing the ability of cells to link up to other cells through pH adjustment. Cells have these type of mechanisms as well. In this case, the cytosis, endocytosis, that allows it to also move without us manipulating it, if that makes sense. These, during diffusion of paracrine factors, paracrine factors like WNT are affected by different proteins. So there's a WNT, a WNT protein called wingless, and so it's called wingless because the mutant of this results in Drosophila that don't have wings. So that's where it gets, a lot of these get their names from mutations from Drosophila. And so there's another one, protein called SWIM. And so if you have SWIM, it's a protein that stabilizes that wingless protein or paracrine factor and helps it to diffuse a further distance. So when SWIM is not present, if you get rid of it, it does not disperse and is kind of confined into this narrow band, right? So wingless is the actual, WG is the actual paracrine factor. SWIM is kind of a chaperone. So it helps it to diffuse, and it helps it to not break down, kind of prevents it from, or it helps it, enables it to get to the spot it needs to get to. So if you get rid of SWIM, then we see it's not like spread out nice like in this. It instead is confined to this narrow band. So similarly to wingless, wingless usually activates another gene called distalless. And again, distalless, which is activated by wingless, if we see it normally when you have SWIM, is a nice expression. However, if you get rid of SWIM, again, that is messed up. So these paracrine factors interact with other proteins and with themselves to build these gradients. So it's not just the paracrine factors that are there to cause the proper gradients. They also have chaperones, or helper proteins, we'll call them. Let's see. So we're going to get into FGF specifically coming up, but FGF is fibroblast growth factor. So secretions of this protein show a comprehensive example how HSGP, which is heparan sulfate proteoglycans, can influence paracrine factor diffusion. So FGFs can interact with these HSPGs. Let's see, do we have that even listed on here? No. Let me see. Can I do a pen? So this is gonna be bad because I am not good with HSPG. Okay, so FGFs can interact with HSPGs, which I said were heparan, HEPARAN, sulfate proteoglycans, and this can influence this diffusion as well. So when FGFs interact with these HSPGs, they either can modulate, reduce, or increase the diffusion. So we'll look at this in, let's see. Let's go to the next slide here. So the quantification of these FGF, this is FGF8. So this is another paracrine factor, and we'll look at binding, like I said, in the next coming lectures. So this normal FGF concentration, we have the control, which is without one of those HSPGs, and you can see the distance that it travels. However, if you add dynamin, which is one of those HSPGs, we see that you get a greater diffusion. So the concentration is higher for a longer distance. Similarly, I can write on here, this RAB is the opposite. So it kind of binds it up, and it does not allow as much at that. It reduces the diffusion ability of that FGF. So let's look here. So what are some of these mechanisms for determining this gradient? Like we just talked about those proteins. Where's my pointer? Okay. Here, I'm going to turn back on pointer here quick. Pointer. There, I can see that better. So there's 5 primary mechanisms for shaping this FGF8 gradient. And it's important for establishing different tissues, right? So we've talked about gradients are very important for these tissues. So one way is the difference in transcription. This coupled with how fast the RNA is decaying before it gets transcribed into a protein. The second is that determines this gradient is the free diffusion. So the closer you are, the higher the concentration, the further away, the lower concentration. So the amount you produce, the distance away you are 2 of the factors that affect the gradient. There are these HSPG directed diffusion. So these fibers here, these HSPGs allow diffusion or can manipulate diffusion, right? So they can travel rapidly along these HSPGs to diffuse faster. In contrast, however, there's some really high density areas of HSPGs that can also reduce the ability. So in this case right here, if we have like these little highways built by HSPGs, they'll diffuse fast. And we're not going to get too much into the mechanisms of this, but if there's orientation for quick diffusion, HSPGs can happen, or this tangled web of mess that can jam up the FGF8 and cause slower diffusion. It's a little confusing. And lastly, there is, let's see, where is it on here? The FGF8, remember that's this guy, FGFR complex can also be internalized by endocytosis and targeted by lysosomes for degradation. So they can also, that is, let's see, is it 5? So this FGF8 can be taken in by cells as well and degraded to reduce the concentration across, as opposed to, so normally, so in this case, look, how much, the best way to put this, think of it this way, the amount of FGF here, the concentration this cell experiences, can be affected by this FGFR complex, where instead of expressing the gene like these ones are doing, it can be just broken down in the cell. So even though it makes it to the cell, in terms of how much this cell is expressing gene-wise is reduced, because some of the stuff that the ligands that are binding are being degraded and not actively expressing or causing transcriptional changes. Okay. Last slide, where are we at? 20 minutes. Okay, this is a bit of a long run. So this is the last slide for paracrine factors. So an important point here is that receptors are not everywhere on the cell. There's small protrusions called cilium, not to be confused with cilia, that hosts some of these. So in this case, hedgehog receptors are on these little cilium that point out here. So you can see here where we have this overlay of hedgehog being expressed. No hedgehog binding here, and then once hedgehog is hyperactivated, we see this change. Don't get too much into it. Just know that there are sometimes these little cellular features that host some of these membrane proteins. They're not all transmembrane proteins in the sense that they're completely surrounding the cell, like cadherins might be. Some of these are on these cilium that are kind of reaching out to feel for paracrine factors. Okay, that is it. 21 minutes. That is it for this lecture. We'll talk a little bit more in depth about like WNT and FGF and hedgehog and some of those in the coming lectures here. So I'll catch you in the next one.